Okay, good morning, everybody. It's not on. All right. Okay, let's get started. So, in last class, we finished the metabolism of nitrogen, right? So, of amino acids. And today we're going to talk about the carbon metabolism of amino acids. So for nitrogen metabolism that we learned from last class, that we usually need to remove the nitrogen first, right, in order to completely metabolize amino acids for energy use. So the nitrogen, and there are several ways, right, the several reactions that involved in the removal of nitrogen. Do you guys still remember? What are the reactions? Transamination and deamination, right? And also one reaction that also important, not for the most of the amino acid, but it's also important. Remember which reaction? Glutamine synthesis, right? So all these reactions, the purpose of these reactions is to remove nitrogen from amino acids as a compound. What is a compound? Nitrogen that removed from amino acids as ammonia, right? And how ammonia is eventually removed? Urine cycle, right? So urine cycle is very important. Okay, now once once the nitrogen is removed from amino acids, then the carbon still kind of. So carbon chain of amino acid then be further metabolized. So here we just go through sort of overall catabolism or metabolism of carbon chain of amino acids. First of all, the rate of amino acids catabolism is determined by amino acids abundance in the body. So basically in free amino acid pool, if you still remember. Oh, it's so noisy. Oh, I should raise my voice, right? So, okay. So amino acids catabolism increase when amino acids are abundant, right? When we have lots of amino acids that we need, for example, after meal. That we have absorbed a lot of amino acids from the diet, right? Under this condition, amino acids catabolism is increased. And also under the hypercatabolic state, for example, uncontrolled diabetes, for some unknown reason that they have lots of protein that released in the body tissues into the circulation or the free amino acid pool, and also under the starvation, right? We need energy, alternative energy fuels, and so particularly under the prolonged starvation condition, the amino acids will be also used for energy, particularly for the glucose synthesis, right? For gluconeogenesis, to maintain blood glucose level at the under the fasting condition. So so again, after meal and also during starvation or uncontrolled diabetes condition. So all these three conditions that you have increased amino acids in the pool, so that you will amino acids will undergo the catabolism. And the other condition, when we have inadequate amino acids or insufficient amino acids, for example, we consume a low protein diet, right? Then our body needs amino acids for synthesized proteins. So under this condition, the catabolism amino acids will be decreased, right? Will be inhibited. So we wanted to spare or save more amino acids for protein synthesis, right? So, for example, high, I listed two conditions, like high fat, low protein diet, right? So you have adequate energy, but you have a low percentage of protein in the diet, or high carbohydrate and low protein diet. But under this condition, that we need to spare protein for protein synthesis for maintain of a physiological function of in the body, right? Because lots of important enzymes and hormones and immunoglobulins, right? They are all proteins, right? They need to be maintained at the normal levels. Okay, so this slide show us the overall pictures of metabolic fate of amino acids. Again, depend on the availability of amino acids in the pool, right? And also depend on the fasting condition, like energy metabolism. So amino acids can be metabolized differently. So, for example, so under the fat condition, if we have an adequate intake of a carbohydrate, right, that means we have enough calorie intake, then the question is also depend on you, right? You know, whether amino acids will be catabolism or catabolized or anabolism, right? Sorry. So if we have enough amino acids, right, then we need for protein synthesis, then the amino acids will undergo catabolism, right? So the two parts, right? One is the nitrogen metabolism and carbon metabolism. So first we need to remove nitrogen as the ammonia, and ammonia will be detoxified by urine synthesis, right? Synthesized urine. And the carbon of the amino acids will go oxidation first to remove alpha keto acid, right? And alpha keto acid can be further oxidized to produce ATPs. And again, if we have more amino acids than we need for protein synthesis, and some of the excessive amino acids will be used to convert to fatty acid synthesis, right? And also for gluconeogenesis. But under this condition, the glucose synthesized from amino acids through gluconeogenic pathway will be converted to glycogen stored in the body, right? Does that make sense? So I just want to point out here the gluconeogenesis not only occur in the fasting condition, but also in the fat condition, particularly when we have larger consume a larger amount of protein amino acids. That amino acids will be converted to glucose and then stored in the form of a glycogen, right? So and the second question is, if we don't have enough amino acids for protein synthesis, right? So in the body, if we have a lower free amino acids in the pool. And under this situation, then the amino acids will be used for protein synthesis, right? So under the fat condition, it also depends on the abundance of amino acids in the body, whether we have abundant amino acids or insufficient amino acids, then amino acid will be metabolized differently, right? And also under the fat condition, if we don't have enough calorie intake from carbohydrate, then amino acids will be catabolized, used for synthesis of glucose under the fasting condition, right? Like similarly, like under the fasting condition. So now let's look at the fasting condition. So under the fasting, amino acids will be used for catabolism, right? So first of all, nitrogen needs to be removed as ammonia and synthesized for synthesis of urea or excretion, like get rid of ammonia. And amino acids carbon will oxidize to alpha keto acid, right? And for making glucose and ketone bodies under the fasting condition, right? So the gluconeogenesis during fasting condition is to make glucose and release into circulation to raise the blood glucose level under fasting condition, right? Which is different from under the fat condition. Glucose will be converted to glycogen and stored in the liver, right? Any questions about this overall metabolic fate of amino acids? Again, depend on fasting condition, and also depend on the abundance of amino acids in the body, right? So can undergo different metabolic pathway of amino acids. And also you wanted to remember that gluconeogenesis also occur in the fat condition, right? The precursor would be the amino acids, right? But for the synthesis of glycogen under fat condition, but on fasting condition, gluconeogenesis from amino acids, the purpose of this was to produce the glucose and release into circulation, right? Question? Okay. Yeah.
Why is urea produced in both fat and fasting?
Sorry, what you said?
Why is urea produced in both fat and fasting condition?
Oh, why? Okay. So because the carbon chain of amino acids metabolism requires the removal of nitrogen, right? So the amino acids undergo oxidation in both fat and fasting condition, right? So the removal of nitrogen is needed. So that occurs in both fat and fasting condition, right? Any other questions? Okay, yeah, that was a good question, sure. Okay, and if you still remember the TC cycles, right, that actually involve a lot of amino acids metabolism, and this here is the sort of summary of a TC cycle that involve in the many amino acids metabolism, right? Metabolism of many amino acids. Like glutamate, all this group of amino acids can be converted to glutamate, right, and then the alpha ketoglutarate, and just different group of amino acids can be metabolized to the intermediate of the TC cycles, right? And we also mentioned earlier that gluconeogenesis pathway that involved the part of the TC cycles, particularly when we use the amino acids as the precursors for gluconeogenesis, if you still remember, right? And some of the amino acids, for example, leucine and lysine, they metabolize to the acetyl-CoA, right? So that means these two amino acids are only ketogenesis, and they are not able to be used for gluconeogenesis. And the other amino acids that they can be used for gluconeogenesis because they are able to enter the intermediates in the gluconeogenic pathway, right? And acetyl-CoA, once it's entered the TC cycle, will be completely oxidized, right? Acetyl-CoA only has 2 carbons, right? We mentioned earlier that it's not precursors of a gluconeogenesis, right? So again, the leucine and lysine are the ketogenic amino acids. They only be able to do the ketogenesis for synthesis of ketone bodies, but not gluconeogenesis, glucose synthesis, right? So carboskeleton of amino acids usually are metabolized to glycolic and TCA intermediates, right? Just I showed you in the last slide. So a lot of amino acids actually be able to use as a precursor of glucose, so gluconeogenic amino acids. So we call those amino acids glucogenic amino acids, right? And some of the amino acids that we mentioned, right, the leucine and lysine, they are metabolized to acetyl-CoA, right? So these amino acids are the ketogenic amino acids, right? So they're pretty easy to remember, right? Leucine, lysine are the ketogenic amino acids, and the other amino acids are glucogenic amino acids. And any amino acids that is potentially glucogenic is also potentially ketogenic, but not vice versa, right? So ketogenic amino acids is not able to do gluconeogenesis, but glucogenic amino acids is also be able to become a ketogenic, right? Does that make sense? Okay. Sorry? Yeah, from acetyl-CoA, right. Okay, so more information about the catabolism of amino acids. So liver is the major site for amino acids catabolism or oxidation and the urea production, right? So liver can do a lot of things. And small intestine, the glutamine catabolic oxidation. So small intestine is the major site for glutamine metabolism, right? We're going to talk about a detailed in later, right? Talk about specific tissue, amino acids metabolism in specific tissues, right? And muscle catabolism of branching amino acids. Muscle is a major site for branching chain amino acid metabolism, right? Like leucine, isoleucine, valine are the branch chain amino acid. So the lots of amino acids that are not used for protein or peptide synthesis in the liver, right? So in the liver, most of the amino acids were not used for synthesis of protein or peptide. Or in other words, the protein synthesis activity is not very active in the liver, right? So most of amino acids are partial metabolized in the liver to generate alpha keto acid, right? And also can be used for gluconeogenesis, right, in the liver for producing glucose or glycogen, right, depend on the abundance or availability of amino acids in the body. So amino acids, again, we also mentioned, right, some of amino acids are the glucogenic and or also the ketogenic. So amino acids can be metabolized to synthesize ketone bodies, fatty acids, right, in the body. So in the liver. Okay, I just want to summarize again, right? The amino acids in overall, in general, the amino acids metabolized in the liver. So liver, again, is a very important tissue for amino acid metabolism, and most of the amino acids will only partial oxidize to generate alpha keto acid, right? And will use as energy in the liver. This first point. And second point, that amino acids can be metabolized in the liver to synthesize glucose through gluconeogenesis, right, and glycogen. And third point, the amino acids can also be metabolized to generate fatty acids and ketone bodies, right, in the liver. So, and last point is that the protein synthesis is not the main pathway in the liver, right? Any questions? And muscle is a major site for branch chain amino acid metabolism, right? And small intestine is a major site for glutamine metabolism. So unlike the other tissue like a brain, right, or red blood cells, mostly glucose are used for energy, right, or ketone bodies. And liver, the amino acids are the important energy fuels for liver and small intestine and immune cells, right? So 50% of ATPs that are needed for the liver actually is from partial oxidation amino acids, from alpha keto acid, right, I mentioned. So 50% of ATP is generated from alpha keto acid, which is initially metabolized from amino acids. And in the small intestine, actually 80% of energy fuels are from amino acids, particularly the glutamate, and aspartate, right? And in the muscle, amino acids are not the primary energy fuel for the muscle, although muscles store a lot of protein, right, and also break down protein during the fasting or the starvation condition, but muscle normally muscle cells normally don't use amino acids for energy. But what are the main energy fuels for muscle, skeletal muscle? You guys know. Glucose and fatty acids, right? And not amino acids. So again, different tissues use different type of energy fuels. Just you know the information. All right. Any questions? So these two slides are just the overall pictures of amino acid metabolism as energy fuel and how they metabolize, right, in the different tissues. Okay, so this slide, together with the next slide, we're going to talk about the metabolic flow of amino acids in different tissues. So different tissues metabolize amino acids differently again, right? So under the fat condition, and this slide shows the fat state, you see the muscle, liver, intestine, and kidney, they are all involved in amino acid metabolism, right? But they have a different role in amino acid metabolism. So in the small intestine, basically the intestine absorbs a lot of dietary amino acids, particularly glutamine, glutamate, aspartate, and branched chain amino acids, right? So absorb lots of this type of amino acids into the portal vein, and the 20% of the branching amino acids will go to the liver, right? But liver is not the main site for branching chain amino acid metabolism. Muscle is the main site, right? So the 60% of a branching chain amino acids will not metabolize in the liver, but goes to the muscle. From liver go to the muscle for metabolism, right? So muscle takes up actually lots of branching amino acids and metabolize there, but release the glutamine and the alanine, right? So the kidney will takes up glutamine and releases the serine and alanine, right? So alanine basically released from muscle, intestine, kidney, and eventually metabolized in the liver, right? If you still remember that the one of the important cycles, gluconeogenesis cycles, is alanine cycle, right? So alanine takes up from the circulation by the liver and converted to glucose in the liver. So three point here is that dietary amino acid patterns. For glutamine, glutamate, aspartate account for 20% of a dietary protein. So dietary protein contains a lot of glutamine, glutamate, aspartate, right? And circulating amino acid patterns, in the circulation, there's high levels of alanine and low levels of glutamine, glutamate, right? And the branching chain amino acids actually increased after a high protein diet. Question? Yes.
So what are the percentages in the diagram? Is that 60%, like 60% of the 20% that's going into the liver?
Yes. Good question. Right. like, the 40% of this 20% metabolized in the liver, but 60% actually goes to the muscle for metabolism, right? All right. So just to tell you, the different tissues that they metabolize amino acids differently, right? And which amino acids are increased in the circulation during fat condition. You probably wanted to know that the branching amino acids are increased after meal during the fat condition. Now, what about under the fasting condition? So fasting condition, you see that alanine will be very high, right? And all the alanine that from the intestine, from muscle and kidney will all go to the liver for gluconeogenesis during fasting condition, right? And produce glucose are released into circulation, and also because of urea will be also synthesized because of high levels of amino acids catabolism, right? So muscle will contribute the glutamine and alanine to the circulation, and the glutamine will be picked up by intestine. So intestine, if you still remember, that utilize a lot of glutamate for energy, right? So during fasting, the intestine pick up glutamine for use as energy, and the kidney will also pick up the glutamine. It is a form of a ammonia transport, right? When we talk about the transamination, deamination, and removal of ammonia, if you still remember, the glutamine synthesis is very important, right? Glutamine has two nitrogen amino group, right? And glutamate is one amino group. So the glutamine synthesis is a way of the removing the ammonia, right? So ammonia being picked up by the kidney, and then deamination, right, and release the ammonia. So this is a way of the release of the ammonia in the kidney. Right. So yeah. Any question? So you guys probably pretty familiar with this diagram, right? The alanine cycles is the gluconeogenic cycle, right? Gluconeogenesis pathway. So alanine, that is the precursors of gluconeogenesis and mostly released from the muscle. So amino acids metabolism in starvation depend on the stage of the starvation, whether it's the early stage versus or the later stage, right? So amino acids can be metabolized differently. So during early starvation, the main purpose of amino acids metabolism is to maintain blood glucose level, right? So we use a lot of amino amino acids for gluconeogenesis to raise the blood glucose level during the early stage of starvation. And of course, in addition to break down the glycogen, right, we also need to break down protein to synthesize the glucose from amino acids. And later starvation. So during the later starvation, because of a lot of amino acids will be break down for gluconeogenesis during the early stage, right? So we lose a lot of proteins. So under this condition, then our purpose is to spare amino acids. We don't allow the protein breakdown further, right? So we don't want to further break down proteins. So under this later stage, our body will use more energy fused from fatty acids, from ketone bodies, right, and try to spare the amino acids catabolism. So again, the different stage, early versus later stage, the amino acids can be metabolized differently. First stage, we'll use amino acids for gluconeogenesis, and later stage, we spare amino acids, so reduce the gluconeogenesis from amino acids, right? Like alanine cycle will be reduced during the later stage, right? Try to spare amino acids, and ketogenesis will be increased, right? And to use as an alternative energy. All right, so regulation of amino acids, oxidation, gluconeogenesis, and ureagenesis. So these three metabolic pathways primarily occur in the periportal cells in the liver. So liver has a different type of hepatocytes, right? I think probably in the couple slides later, we're going to show you the location of the different type of hepatocytes, and the metabolize the amino acids differently. And these three pathways, amino acid oxidation, gluconeogenesis, and ureagenesis or urine cycles, these three pathways actually interrelated, right? They are regulated, coordinately regulated together. So these pathways are all active in both fat and fasting state. So the concept may be a little bit different from what we learned from the glucose and fatty acids metabolism. Normally we thought catabolism will be active during the fasting condition, right? And anabolism mostly active in the fat condition. But amino acid metabolism is different, right? The oxidation, gluconeogenesis, and urine cycle all occur in both fasting condition, right? But again, the amino acids availability is most important. That regulates, whether they undergo which metabolic pathways, right, of amino acids. Do you guys want a break? Okay. let's move on. So this slide is the overall picture of amino acids metabolism in the liver. So starting from this slide, we're going to talk about amino acids metabolism in specific tissue, right? So this is the first tissue organ, liver. So how amino acids are metabolized in the liver. So this again is the overall picture. In the fat condition, so lots of dietary amino acids, right, be absorbed by the small intestine and to the portal vein and to the liver. So about 60% of the dietary amino acids are metabolized in the liver. 20% of them used for protein synthesis, and another 20% of dietary amino acid that actually released into the circulation and for catabolized in other tissue, right? So this is the sort of overall, the idea about the amino acids metabolized in the liver. So it's actually a lot, right? 60%. A lot of amino acids are metabolized in the liver. And more information about AA metabolism in the liver. So again, about 60% of amino acid that absorbed and transported into the liver are catabolized in the liver, right? First point. And in terms of a metabolic pathway, or how amino acids are metabolized in the liver, the first is nitrogen. So nitrogen, again, always need to be removed as ammonia. And ammonia need to be removed, otherwise it will be toxic to the body tissue cells, right? So the removal of nitrogen or ammonia by the ureogenesis or urine cycle, urine synthesized urine, and then excreted in the urine, right? And carbon skeleton of amino acids can be metabolized through the several different pathways. First of all is the oxidation. So amino acids, skeleton of amino acids oxidize to alpha keto acid, right? And also the carbon skeleton of amino acids can be used for gluconeogenesis, for produce glucose released into the circulation during fasting, or for glucose, for produce glucose that being converted to glycogen under the fat condition, right? But sorry, I did not list it here. Actually, one more point, one more pathway that amino acid can be catabolized is for ketogenesis, right? So ketogenesis. So which are the amino acids that only be able to do the ketogenesis? Do you remember? Leucine and lysine, right? So these are the two ketogenic amino acids. But of course, the other glucogenic amino acids can also be used for ketogenesis, right? Okay, so liver anatomy. So liver has a central vein or hepatic vein and also portal vein, right? And the artery and other important system like bile acid, right? Secretion system like common bile and duct and also gallbladder, right? So this just vessel system in the liver. They have a different from the other tissue organs. Have artery and also vein, but liver has a two different set of vein, like central vein and portal vein. And this the liver cross section. So the cells that around or nearby the portal vein called periportal hepatocyte, right? And other set of cells, or group of cells, or type of cells, whatever called, that nearby portal vein is called a perivenous hepatocyte. So perivenous hepatocyte and periportal hepatocyte, they are two different type of liver cells, and they do a different metabolism, right, in amino acids. So they have a different location. So just you know that the two different type of hepatocyte. So urine cycle is very important. The amino acid metabolic pathways in the liver, right? So liver is the only tissue that does 4 cycles of urine, the 4 urine cycles. So the purpose of a urine cycles is to remove ammonium, right? Which is generated from transamination, deamination, right? So so this just show you the whole process. The amino acids undergo the transamination by the set of enzyme called aminotransferase, right? Depending on which amino acids that involved in this reaction, and the deamination, the glutamate dehydrogenase. So all these 2 different type of reactions eventually will generate ammonium, right? So remove nitrogen from the amino acids and release as the ammonium in the circulation. So ammonia is toxic, right? Again, to the tissues, organ, particularly to the brain. So we need to get rid of the ammonium, and urine cycles is most important pathway that for the removal of the ammonium in the body. Any questions? Okay, now let's get to the details of the urine cycles. So urine cycles involve 5 steps, and in 2 different intracellular compartment, right? Mitochondria and cytosol. So if you still remember, we just mentioned a little bit, right? The overall pictures of a urine cycle earlier. The first 2 steps occurs in the mitochondria, and the rest of 3 steps in the cytosol. And also very important information you probably wanted to remember is that urine cycles primarily occur in the periportal hepatocyte, right? And not in the perivenous hepatocyte. So now let's talk about the first 2 steps in the mitochondria. So the substrate for urine cycles, right? One is ammonium that we need to get rid of, right? It's generated from the transamination and deamination of amino acids metabolism, and the carbon that from the CO2 or bicarbonate, right? So ammonia and CO2 or bicarbonate together form the carbamol and phosphate. So this is the first step, right? First reaction. So this reaction requires enzyme called carbamol phosphate synthase, so CPS1. So CPS1 is the enzyme that its activity requires the NAG, NAG, and magnesium also required for the enzyme activity. So NAG is the N-acetylglutamate, right? So this compound is required for the activity of CPS1 for the first step of a urine cycle for generating carbamyl phosphate, right? So carbamyl phosphate is generated from one molecule of ammonium and bicarbonate, right? And the second step, the carbamyl phosphate together with ornithine form citrulline, right? So citrulline is generated in the second step from ornithine and carbamyl phosphate. And the enzyme for the step 2 is ornithine transfer carbamylase, OCT, right? OCT is the key enzyme for the second step. So these two enzymes are very important, right? For the first two steps of urine cycles in the mitochondria. So the product of the first two steps is a citrulline, right? So citrulline, once it's generated in the mitochondria, then released into the cytosol for the rest of three steps reactions, right? So something that you need to remember for this first two steps urine cycle in the mitochondria. First of all, the urine cycles takes place in the periportal hepatocytes, right? And second, the first two reactions occur in the mitochondria. Ammonium comes from deamination of amino acids, all right? So this ammonium, first ammonium, is coming from deamination of amino acids. Why we need to do the deamination? Again, I repeat many times, right? Because in order to metabolize carbon, we need to remove nitrogen, right, from the amino acids. And step 1 uses the 2 ATPs, right? So 2 ATPs are needed for step 1. And NAG, N-acetylglutamate, is a compound that required for step 1 for CPS1 enzyme activities, right? So all these points that you probably wanted to remember to help you understand, the first two steps of the urine cycle in mitochondria. Okay, now step 3, which occur in the cytosol, right? So once the citrulline is synthesized in the mitochondria, released into the cytosol, and together with aspartate. So aspartate carries the one amino group, right? So so aspartate actually provides the second amino group to the molecules, right? To the urine cycle, to the urine molecules. So citrulline and aspartate together form arginosuccinate, right? So arginosuccinate has the two amino groups, one from initial step, right? Initial reaction from actually amino acid deamination, right? And the second amino group is from aspartate. So in the step 3. So the step 3 need this enzyme, right, for the activity, for the reaction. So you also wanted to remember this 3 points here. So step 3, 4, 5 takes place in the cytosol, and step 3 uses the 2 ATPs. right? So step 1 uses 2 ATPs, and step 3 also uses 2 ATPs. So now we need 4 ATPs, right? At this point. And the third point you wanted to know is that second amino group is transferred to citrulline from aspartate, right here. So now we have a 2 amino group, because urine, if you remember, we mentioned, has a 2 amino group, right? So one initially from the ammonium, and the second amino group is from aspartate. All right, step 4. So arginine succinate, once it's formed, then it's break down to arginine and fumarate. And this is an enzyme called arginine succinase, right? It's an enzyme catalyzed as the step 4. So arginine, once it's formed, arginine carries again the 2 amino groups, right? Okay, so the last step, step 5 in the cytosol, is the formation of a urea. So urea, you see these are urea molecules, right? It has 1 carbon, 1 oxygen, and 2 ammonium groups. And arginine can be hydrogenate to generate urine, right? To adding the oxygen. So oxygen basically is from the water. The arginase is an enzyme to catalyze this reaction, right? And arginine, once it's released the urine, then they convert it back to the ornithine. So ornithine gets recycled, right? In the last step, then we'll go back to the mitochondria for the next cycles of urine synthesis, right? So any questions about the urine cycles? Right. So several key points you wanted to remember: which type of cells that urine is synthesized. Peripodal hepatocytes, right? And 5 steps, 2 steps in the mitochondria, 3 steps in the cytosol, and 2 steps in the mitochondria for generating citrulline, right? And key enzymes, the CPS1 and OTC, are the 2 key enzymes in the mitochondria for urine synthesis, and the regulator compounds like NAC, right, and acetylglutamate is required for urine synthesis, or CPS1 enzyme activities. And also important thing that you need to remember, the first steps, the substrate, one is ammonia, right? And the other one is bicarbonate, CO2. So ammonia, the first ammonia is coming from deamination of amino acids, right? So that is a purpose, right? We want to get rid of ammonia from the nitrogen metabolism of amino acids. And a second ammonium or amino group is from which amino acids? Aspartate, right, in the cytosol. So again, because urine has 2 ammonium groups, so one from the ammonium, basically from the deamination of amino acids, and the second ammonium group is from aspartate. And ATP, uses of ATP, so far 2 steps, right, that requires ATPs. First step requires 2 ATPs, and the third steps requires also two ATPs, right? So 4 ATPs is required for urine cycles. But the fumarate is generated in the urine cycle can also be recycled, right? So this is a process for the recycle of fumarate. So fumarate being converted to malate, and malate dehydrogenase and converts malate to oxaloacetate, and then to aspartate, and back to the urine cycles, right? And during the dehydrogenase reaction, malate dehydrogenase reaction, the 1-NADH is generated. And if you still remember, 1-NADH usually produces 3 ATPs, right? So during the recycling of a fumarate process, the 3 ATPs are generated that can be used for urine cycles. So the net use of ATP or need of ATP is how many ATP for the complete of a urine cycle? Size 1, right? Yes. Because 4 ATPs are used during the step 1 and step 3, right? Reaction. And 3 ATPs are generated during the recycling of fumarate. So net need of ATP is 1, right? Does that make sense? Okay. Okay, now let's talk about intercellular grooming cycle in the liver. Right, now we're still in the liver, right? We talk about the 2 different types of hepatocyte, right? Liver cells, periportal hepatocytes and paravenous hepatocyte. So these 2 types of cells, again, have a different function, play a different role in the amino acids metabolism, particularly in the nitrogen metabolism, right? So periportal hepatocytes basically has a very important function in the urine cycle. Urine cycle occurs primarily in the periportal hepatocyte, right? So again, urine cycles, the purpose of urine cycle is get rid of ammonium. So ammonium basically is generated from the deamination of amino acids, right, in the circulation and picked up by the periportal hepatocyte and enter the urine cycle, right, for synthesis of urine and then released into circulation and excreted in urine, right, urine. And grooming, I mentioned, is also a transporter of ammonia, right, from other type of cells that can be delivered to the periportal hepatocyte. So undergo the reaction called the glutaminase, right, to grooming being converted to glutamate and release one of the ammonia. And glutamate be further undergo deamination and release another ammonia group, right? So eventually all this ammonia, either from the circulation, from the other tissue cells, deamination, or the grooming, right? The glutaminase or deamination from the glutamate, then be enter the urine cycle, right, again. So this is something happening in the periportal hepatocytes. So periportal hepatocyte is the main or most important cells that does urine cycles and remove the ammonia, right? And paravenous hepatocytes serve sort of a backup mechanism, right? Backup place. So, for example, some of the ammonia that has not been picked up by periportal hepatocytes that leak, right? Then the paravenous hepatocyte will pick up the leaked the ammonia, and then do the grooming synthesis reaction. So the ammonia that's been added to glutamate and forming glutamine. So glutamine again has a 2 amino group, and glutamate only has a 1 amino group, right? So this is a glutamine synthetase reaction is very important, right? It's a way of removing ammonia, right? That serve as a backup mechanism, right, to remove the ammonia in the circulation, right? Any question about this intercellular grooming cycles in the liver? So again, 2 different types of hepatocytes does a different thing. So the periportal hepatocyte is the main cell that remove ammonium from the circulation through the urine cycles, right, urine synthesis. And paravenous hepatocytes just serve as a backup mechanism to pick up the ammonia that has not been, or cannot be picked up by periportal hepatocyte. And the mechanism is also different, right? In the paravenous hepatocyte, the glutamine synthesis is the mechanism to remove ammonium, right? Okay, any questions? No? Okay, so, and we have, let's see, the critical thinking, right?